Thank you, Jan, and it's a great pleasure to be in the meeting again and to see the development. Now, I shall try in very few words to say a little bit about what INCF can do for neuroscience. Of course, you have heard during the last 2 days a lot of the activities of INCF, so I would be rather brief, but let me just say first that why do we need neuroinformatics? Neuroinformatics is, of course, one part of neuroscience, one way of approaching neuroscience and to understand the brain, and I would say an indispensable part. The reason is, of course, that you have, on one hand, you have had a very dramatic development on the molecular side, signaling pathways, how single neurons operate. You have had an astounding development on the imaging side. But of course, you need to marry these if you are to account for behavior in terms of nerve cells, transmitters, and so forth. And in between these two levels, you have the levels of systems and microcircuits. In order to understand behavior, you really need, in a systematic way, to bridge from this side to that side. You need to bridge from gene and gene products to their role in cells, synapses, networks, and behavior, cognition, etc. There is no way around that you really need to have the different links. It's easy to say, very difficult to achieve. You can have the role that a given gene produces a given disease, like Huntington gene. So one knows that a gene produces a particular phenotype, maybe a malady. But in order to not only know that it's causing this, but to know why, one needs to have the link in between. There is no way around that. Maybe it's not so important sometimes, but I mean, with all aspects of behavior, you really need to understand this. And on the other hand, to understand dynamic processes, you need modeling, because our brains are not able to handle sort of complex dynamic processes where you have a lot of interacting factors. In order to be able to model and try to model parts of this, of course, you need to have access to all the available data on the different organizational levels. And that's where databases becomes absolutely fundamental. And the average researcher hopefully knows his own level in the organizational scheme, and perhaps plus minus one level, but that's normally what a given researcher can bridge. And in order to be able to pick up relations between genes and synapses, or synapses and complex cognitive function, you need to be aided by the available data, and that's available in databases. So these are the two different things. Essentially, if you look in a sort of a little bit more granularity, from gene to behavior, you of course have gene and gene products and how they are expressed. And you have the next level, is called the molecular machines, that are much more complex to understand. If you take the postsynaptic density with more than 100 proteins that are hung up in a particular organization, we need to understand how come and what sort of they achieve. You can say that the spines of individual neurons are small molecular machinery for synaptic plasticity. We need to understand how they operate, and of course all the signaling pathways. And you have the level of synaptic function, modulation. You have the next level, sort of small integrated circuits, that I think is also absolutely critical. If we are to understand cortical function in one day, we have to start with understanding how a cortical column operates, and then of course how they talk to each other. We have the multi-pattern generator circuits, the CPGs, we have striosomes, we have cerebellar microsomes, and there could be long lists. So that's one level. And that sort of is the cellular molecular underpinning
of what goes on. Then that translates into integrated functions. You have action, and action, of course, is perception, evolution, motor programs that are required. You have the learning and memory with storage and retrieval. Storage, I think we perhaps start to some degree to understand. The process of retrieval is much more difficult. We have the emotions, how our brains, the brains of any animal, interprets a given stimulus to give it an emotional color. And we have the internal emotional representation, sort of how we feel when we are angry or glad. And then you have the motor programs underlying emotions. Humans have about 7 motor programs, each of which displays a given anger. So when we smile, when we laugh, when we cry, etc. And of course you have the most complex aspect, cognition, selective attentions. And that's the normal function of the brain, the sort of a few of the different aspects. And then neuroinformatics is also important for society in terms of medicine. Of course, 1/3 or a little bit more of the costs for health care in North America, Japan, or Europe is due to diseases of the brain, since most of them are chronic. You have the importance of the brain in education, training, and of course towards the technical side, information processing, robotics. So it's a fairly broad range where one needs to understand the brain or aspects of the brain. So, and INCF, of course, comes in as providing some aspects of infrastructure for the rapid development of neuroinformatics. One key is modeling, either based on rigorous experimentation that sort of can help interpret the results. One also needs exploratory modeling where one tests different ideas, and interoperable databases, sort of from gene to behavior and disease and so forth. And that's again where we try to provide some input, and then also in tool development for acquisition, visualization, and distribution. We've been doing less of that. So essentially, it's databases, models, and tools that together form the sort of mission of INCF. We have, as you have heard during the meeting, we have so far 4 programs: multi-scale modeling, digital atlasing, ontologies, as you heard from Marianne earlier, and we have data and metadata standards, which will be changed. The name will be changed to data sharing, which is the essential part of that. And the mission in this context is to try to create standards as an independent organization, and it's of course an important mission to see that one likes to have an agreement of standards, so that each different activity does not limit it, sort of the standards vary between. So that's a cooperative aspect of neuroinformatics. We have, as I guess perhaps all of you know, portal development that is rather substantial, and all information from the programs should be available on the portal. And then we have spent rather considerable efforts also with training to see how we can train neuroinformatics for people or students with different background, or senior researchers. If you have a physical technical background, you require different training from if you are trained in neuroscience, neurobiology, basically. So INCF is organized with a governing board from the 16 different member countries, with a secretariat in Stockholm with about 12 people. which has the most important aspect of which is program officers, and we then contribute then to the programs. The programs are what INCF contributes there directly is the program officers that can help facilitating the work of the different programs. The programs have oversight committees and task forces for specific things, and it involves now altogether about 125 different researchers from different parts of the world throughout this. So it's a fairly large part. But they are doing this work of interest for the development, etc. And it's then important to have the program officers help with this. You have the training aspect. We have different services, this portal. We have the software center where we have a lot of software compiled, the Allen Brain Atlas Mirror in Europe, the Vaxxon Space, multi-simulation coordinator, and some different things. So essentially, if we first think about INCF and the database infrastructure, of course we work for interoperability, and there the digital atlasing is one very important thing. We have the ontologies and the metadata and data sharing. And where we don't have a program, but we think about how to process, is of course a problem around sustainability of databases and programs, which is one thing where INCF most likely can help. And then we, of course, have no resources to do everything ourselves, and we do our best to interact with other initiatives. And particular mention here of NIF and NITRIC, the whole brain catalog, but there are a large number of other efforts also. So and in addition, we have the ambition, of course, to coordinate efforts. And in the original plan, it says International Neuroinformatics Coordinating Facility. So one should avoid the application of efforts. And that's where we at least should have contribute importantly. And we try to. We have, and this slide by Ilya Zaslavsky, sort of, it's the concept of how it works. We have the servers, etc. In Stockholm. We have what you could call the INCF hub that talks directly with the resources in different parts of the world. We have the Japanese node here with all the different databases, and we have different other databases. We have the US with NITRIC and NIF and so forth. That talk. One can take an example here with NITRIC. It has an interoperable link to INCF since June 2009 with David Kennedy as one promoter, and with INCF Software Center. And tools registered at one site are present in the other repository, so it goes both ways. With NIF, we have the INCF broker transfer of brain info as one database to NIF, and sort of interaction with NIF, etc., and with the NeuroLex. So this is sort of the general organization. Let me just very rapidly take you through. You have seen this slide already, but the point that where INCF has contributed is to provide a standard space for the mouse brain into which different databases can be fixed, so that one can go between different databases like the Allen Brain Atlas, the Vaxxon, and so forth. And that's much further developed than much of the work of Ilya Zaslavsky. And this is just showing the space. You can see here that this is the Allen Brain space, and so the brain can be computed in any angle and any direction, and you can identify any structure, and you can go between the Allen Brain
and the different atlases. And the Japanese node has developed a number of different databases in a very laudable way. You heard yesterday about the invertebrate database with its 1,500 neurons from 2 species. And so they combine and they are interoperable and they sort of provide input to INCF and is of course available globally. If you take the ontologies, you have seen this slide before, and the intention and what INCF promotes together with NIF and Marianne Mertone under the leadership, it's essentially to represent terminological knowledge in a machine processable form with a concept hierarchy. And that's, you heard a whole talk about yesterday, but that's one other thing. And finally, the data sharing, it's starting with Colin Ingram and with 2 task forces. If we look on the modeling infrastructure, again, it deals with interoperability between simulators. You heard about the developing new language, the 9ML, and also to facilitate multi-scale modeling. And MUSIC, which is the short for this multi-simulation coordinator, serves to allow one to use different simulators that can interact. So one could have one neuron simulated in Moose and another in Python and so forth, and they can interact in real time. It's Nest neuron Mouse, Python compatible, and it has also been used for interaction in real time, provide interaction with a robot. And it's Mikael Djurfelt who has been the prime mover of this, and that has been developed in Stockholm. So that again, saving time for interoperability, saving time and making simulations more efficient, so you don't need to transfer different things. And the multi-scale modeling task force, Erik de Schutter, and is a chair, and Sean Hill has been a task force leader, and the main product is the 9ML that is on the way. And I then thought that I would take the liberty of taking an example of multi-scale modeling from my own work. And this is mainly with Hergen Kotalevsky, and just show how it has been very important in my own work. Essentially, as some of you know, I spent much time looking on and trying to understand the neural underpinning of goal-directed locomotion. Essentially, you have a spinal network with the central programs, etc., which sort of coordinates the motor program. You have a drive from locomotor command centers, you have steering, you have posture, and you have a mechanism to select. And we know a fair bit about this, but I will just take the example from the CPG, the old work that we've done there. Essentially, we have identified which neurons are responsible. We have in their cellular properties, their synaptic interaction from cell to cell, so we have detailed that in considerable effort. Knowing both cells, cellular properties, synaptic properties, you are still, of course, not certain about those things operate. You can make diagrams like this, where we have a synaptic interaction with different excitator and inhibitor interneurons, etc. You can define that certain parts are core, like the excitator glutamate neurons. You can generate burst activity. The inhibitory neurons generate alternation. And you can show cellular mechanisms that are important for regulating burst termination with different ion channels and calcium and so forth. You can detail that. You can make a qualitative description, but there is no way that you can actually test whether your data can account for the findings. So now, many years ago, together with Anders Lansner in the beginning,
we made detailed models of each type of neuron with the action potential, the after hyperpolarization, the different ion channels that we had established was expressed. So these model neuron performed very closely as their natural counterparts with frequency modulation and everything. So we had populations of cells. We then connected the cells with conductance increase, AMPA receptors and NMDA receptors, etc. So we modeled that. When we put that together, we drove the rhythm from reticulospinal levels. We could then establish that we had rhythmic generation. We could also show that we could generate that in the whole frequency spectrum. That does not, of course, prove that this is the way the network operates, but it shows that with the facts that we have available, we can account for the operation. And there's no way that you could do that intuitively. You need to have it formalized if it should be. Now this is what happens on the single cell level, but in reality we have 100 segments, or on the single segment level, we have 100 segments that produce swimming movements of this creature. And we've recently used large-scale simulations with about 10,000 Hodgkin-Huxley neurons with several compartments, 86, with the approximate and segmental connectivity, and 86 compartments, a fair number of synapses. And as I said the other day, when you have these neurons biologically, we know that they are that they have different sizes, and that turns out to be very important because they are recruited at different points. Low threshold neurons are recruited early and others late. So and that provides stability for the networks. And we have depicted the activity by having the neurons color-coded, so blue is hyperpolarized and red is depolarized. And if we look on the 10,000 neurons from above, you can see that we have activity transmitted along the body in alternating fashion, but also with an undulatory wave with the appropriate. So the network can produce that. The network is flexible, so behaviorally the animal can swim backwards. We know that spinal cord can generate backwards also. If we inhibit this little part, we can actually, just by tinkering with that little part, we can reverse the coupling between the two sides. Okay. Then the next level is that we have modeled earlier and now again the mechanical apparatus, the muscles, and with reduced number of segments and a somewhat simplified network, but with the same principles. And we have there added also the tectum and sort of vision. So essentially, with the tactile function. So essentially, if the model lamp receives blue objects, it will start swimming against them, towards them, and red objects it will start to avoid. And that's a little bit, that's So this is a simulated lamprey in simulated water, and as you see here, it avoids the red one and then hits on the blue one, just to make another level. And we have recently, together with a group in Pisa, also developed robots robot lampreys that swim with sort of biological practice. And with actuators that have properties similar to muscle.
So with that, I thought it was legitimate to take an example of sort of showing the different levels from single cell level with the molecular expression. Okay.
Thank you very much for this elegant presentation. So we have time for a couple of questions, even to the last scientific part here, if there would be any.
Hi. So in your physical instantiation of the Lamprey that you showed in simulation, is there any feedback in that system? Is there sensory processing?
Yes. I mean, we have, in that particular one, it was not, but we have in other simulations included the feedback from the mechanoreceptors receptors along the body, sensing the movements, and the lamprey swims, the model lamprey swims nicely through ordinary water. But if you have perturbations like a faster water speed, etc., it will not be able to cope with them. So the sensory input is quite important. And of course in this later we have very simplified visual input also, but that can guide behavior.